Hello everyone, and welcome to today's lesson on genetics. We're going to be talking about what genes are and how we study them. Let's jump right into it. Uh, let's look at the structure of genes. Take this graphic, for example. If you look at the far left, you'll see the cell. Now, if you zoom in a little bit, focusing on the nucleus, you begin to see X-shaped structures. We call these chromosomes. Now, if you zoom in a little bit more, focusing on these chromosomes, you begin to see that chromosomes are made up of DNA. Now bear with me, if you zoom in one more time, focusing on the DNA, you're now at the scale of genes. Genes are functional segments of DNA. Now I know I might have made it seem like we were done zooming in, but in order to really understand the structure of genes, we need to zoom in a little bit further to the level of nucleotides. An easy way to think of nucleotides is to think of DNA as a long chain and nucleotides as the links that make up those chains. Essentially, DNA is made up of nucleotides. We have actually 4 types of nucleotides in our DNA: adenine, guanosine, thymine, and cytosine. And these nucleotides make up the code or language in which our genetic information is written. As we continue discussing the structure of genes, an important principle for us to talk about is going to be the principle of complementarity. Now that might seem like an intimidating term, but in reality it's pretty simple. Essentially it describes when two things fit together, kind of like puzzle pieces. When we look at our nucleotides, we can see that guanine is complementary to cytosine and adenine is complementary to thymine. And all that really means is that guanine matches very well to cytosine and adenine matches very well to thymine. Now I mentioned this before, but nucleotides are linked to each other in chains to form DNA, and the links between those chains are known as a phosphate backbone. Now when you have two complementary chains of DNA, they wrap around each other and form a shape that many of you guys might already be familiar with, the double helix. Now that we've talked about the structure of genes, we can talk about the function of genes, which is to store information. The way that information is stored in our genes is actually dependent on the sequence of nucleotides. What that means is that the specific order in which our nucleotides are linked to each other actually influences the information that's stored. A codon is a term used to refer to a sequence of 3 nucleotides, and it's the smallest unit of genetic information. Now, what exactly does this genetic information code for? It actually codes for our physical traits, such as our eye color, our height, and our hair color. Before we finish our discussion on the structure and function of genes, we should talk about mutations, when the original sequence of DNA is altered, such as nucleotides being switched out, being deleted, or even being damaged. Mutations can be caused by any number of things, ranging from radiation, random chance mistakes when DNA is being replicated, or even carcinogens, which are cancer-causing compounds. The idea of typos or errors in the information that give rise to our physical traits is scary to you. Don't worry, you're not alone. Mutations can actually be deadly if they disrupt the function of critical genes and give rise to things like cancer or abnormal development. However, not all mutations are bad, and mutations are actually critical for the process of evolution, which essentially is the accumulation of a bunch of harmless or helpful mutations. Over time, if you acquire enough of these mutations, you get new species. Now that we've talked about the structure and function of genes, we can move on to discussing how we study them. A major issue that scientists face when studying genes is the issue of accessibility. Genes are actually tied up in the nucleus of the cell, and it's often difficult for scientists to get a good look at them when they're sequestered away like this. So how do scientists get around this problem? Well, stick around and find out. In order to get an idea for how scientists address the issue of accessibility, we need to talk about osmosis, which is a tendency for differences in concentration gradients to equalize when separated by a semi-permeable membrane. Now, the definition alone isn't the best for explaining what osmosis really is, so let's work through an example. Take a look at this figure. On the left, you have an area of low solute concentration represented by a few yellow pentagons, and on the right, you have an area of high solute concentration with a lot more yellow pentagons. When you have areas of different concentrations separated by a membrane, you have what we call a concentration gradient across the membrane. Now the membrane in between these two areas is only permeable to water, which means that the only way for differences in concentration to equalize is for water to flow across the membrane to equalize the concentration on both sides. Now take a moment and try and guess which way the water will flow in order to equalize concentrations on either side of the membrane. If you guess that the water will flow from left to right, then you are correct. Water will always flow from areas of low concentration to areas of high concentration. This dilutes the area of higher concentration, making it equal to the area of low concentration. Before we finish talking about osmosis, there are three terms that we should familiarize ourselves with: hypotonic, hypertonic, and isotonic. These terms refer to the relative concentrations of solute in a cell versus in its environment. To test your understanding of osmosis, see if you can predict whether water will flow in or out of the cell in each of these conditions. In hypotonic conditions, water will flow into the cell. In hypertonic conditions, water will flow out of the cell. And in isotonic conditions, there's no significant difference in water flowing in or out of the cell because there's no difference in concentration. To get an idea for osmosis in action, take a look at these cells. The cells on the left are in a hypotonic environment, while the cells on the right are in a hypertonic environment. If you look at the cells on the left, they're bulging and swollen, and this is because water has flowed into the cells. On the other hand, the cells on the right are shriveled and shrunken, and this is because water has flowed out of the cells. Now, onion cells have cell walls that help keep them supported when they're overflowing with water. But what about cells that don't have cell walls, like our own? You probably have an idea for how scientists use osmosis to get access to DNA, but let's walk through the actual steps that they take. The first thing they do is use detergent to weaken the cell membranes. Once the cell membrane has been weakened, they place the cell in a hypotonic solution. This means that there is more solute inside the cell than there is outside, and water will flow into the cell. After placing the cell in a hypotonic solution, they simply watch and wait. Water will begin flowing into the cell, and because they weaken the cell membrane with detergent in the first step, it will eventually burst, spilling its contents, like its DNA. And that is how scientists get around the issue of accessibility when studying genes. Well, that's all we have for today's lesson on genetics. Thanks for watching.